Chapter 19, Chromosomes and Cell Division. In this chapter, we're going to talk about two types of cell division, the form of the chromosomes, the cell cycle, mitosis, the creation of genetically identical diploid body cells, cytokinesis, karyotypes, and then meiosis. All right, the process of cellular division is going to be a highly regulated one. Life begins with single cells, but in only a very short amount of time, that cell can lead to trillions and trillions of cells through the process of cellular replication and division. Not only can these single cells produce millions and trillions of cells, but they can also give rise to highly specific cells. Even though you start out as a single-celled organism, there are over 200 different types of specialized and individualized cells in your body, each with their own unique function to perform. This process of cellular replication and division is what we're going to be studying in this chapter. Throughout the course of our lives, we will undergo two different types of cellular division, each one with their own purpose. The first type we're going to go through is meiosis. Meiosis is used exclusively for sexual reproduction. This is only going to occur for the production of gametes or sex cells. Only going to occur for gametes or sex cells. Once two haploid cells fuse to become a single fertilized zygote, the cell is going to undergo mitosis. Mitosis is going to be the cellular replication process, which is used to grow from a single cell into a multi-celled organism. So this nuclear division is used for growth and repair. And specifically for growth, it's used when a fertilized egg becomes an embryo becomes a fetus, after birth, as the child becomes an adult, and when a cut heals or a broken bone mends. Those are going to be the situations under which we're going to employ mitosis. Every cell contains DNA. DNA is how we're going to store all of our genetic information. If we're going to grow from a single cell into a complex multicellular organism, we need every cell to be reading from the same set of instructions. So before we can grow and divide from a cell, we need to make an exact copy of the DNA and then pass it on to the next round of cells. So we're going to be talking about the shape and structure and function of DNA. DNA is going to take on several different forms depending on its current function for DNA. When the cell is replicating its DNA and carrying out its normal daily functions, the DNA is going to remain in long, thin strands, loosely coiled, called chromatin. In this form, chromatin is going to be invisible underneath the microscope. When a cell is replicating its DNA, when it's replicating its DNA and carrying out its normal functions, it's going to be in chromatin. During cellular division, however, the chromatin is going to condense down into chromosomes, which are located inside of the nucleus. The purpose of coiling down our DNA and condensing it into these tight little structures is that it makes the DNA much easier to move, sort. So just for example, let's say that we had a big old plate of spaghetti and we needed to separate every single noodle and make sure that we're putting equal portions of noodles on separate plates. So we can either untangle and untangle all of that spaghetti, or if possible, we could condense it down into like 6 raviolis. And then we just have to put 3 on one plate, 3 on another plate. That's kind of what your DNA does. Now, unfortunately, we don't have the ability to magically condense pasta, but it's a lot easier to sort and organize 6 ravioli than to untangle a whole plate of spaghetti. You're a lot less likely to damage the spaghetti. You're a lot less likely to damage the pasta if it's condensed down into a larger, easy to manipulate form. So those are going to be the form or the shape of chromosomes. When we're talking about chromosomes, each species has a characteristic number of chromosomes tucked inside the nucleus. Humans have 46 chromosomes. Just as example, yeast have 32 chromosomes. Potatoes have 48 chromosomes. Fruit flies have 8, goldfish 94. The black mulberry bush has 308 chromosomes. So the number of chromosomes doesn't necessarily denote complexity in an organism. Humans have 46 chromosomes. We have 2 sets or 2 pairs of all 23 chromosomes, 1 set of 23 from each parent. 22 pairs are going to be autosomes, 1 pair are sex chromosomes. So what we have is 23 different chromosomes found in the human body. You get 1 of each kind of chromosome from each of your parents. Now there are 22 pairs of autosomes. Autosomes are chromosomes that do not code for sex. Sex is coded for by the sex chromosomes, the X and Y chromosomes, found over here. Shown here, we have a pair of two X chromosomes, but that could have just as easily have been an X and a Y chromosome. As we've already sort of said, of the 23 pairs of chromosomes, sex chromosomes make up one pair. They're going to determine gender. There are two types of sex chromosomes. An XX individual is going to be a genetic female. An XY individual is going to be a genetic male. Autosomes make up the other 22 pairs, and they are going to determine the expression of most of the person's inherited characteristics. Somatic cells are going to be all the cells in the body except for the egg and sperm cells. These are what are known as gametes. So just like there are two different types of chromosomes, there are two different types of cells in the body. Starting off with somatic cells, these are the body cells. Each somatic cell has two complete sets of chromosomes, one of each type of chromosome. They're going to receive one chromosome from each parent. When we see these chromosomes laid out like this, we call them homologous pairs. Homologous chromosomes are chromosomes that code for the same information, although it might be a slightly different type. So for example, you're going to receive one chromosome number 1 from your mom, one chromosome number 1 from your dad. Together, both of those chromosomes are considered homologous. Now, these two might not necessarily have identical information, but they're coding for the same thing. So perhaps over here on the tip of chromosome 1, you're going to find a gene that codes for hair color. And over here on the tip of chromosome 1, you're going to find that same gene. That same gene is going to be located in the exact same position of homologous pairs. But that information might be different. You might get a gene for light-colored hair from your mom and a gene for dark-colored hair from your dad, or vice versa. So those are going to be homologous chromosomes. They code for the same information, in this case, color of hair, but the color that they code for might be different. So as we've mentioned, chromosomes occur in homologous pairs. Each somatic cell contains those homologous pairs. One chromosome, as I said, is from the mother, one chromosome from the father. These chromosomes have genes for the same traits, and they also have the same banding pattern. This band indicates where certain genes are located. And what we'll find that on chromosome number 1, we're going to see the genes are located at the same loci, meaning that they are found in the exact same location on either chromosome. Now, like I said before, they might be different information, but they're still coding for the same type of information. Diploid cells are going to be cells with 2 of each chromosome. We represent them with the abbreviation 2N, meaning that they have 2, the number of chromosomes. These are going to be our haploid cells. Haploid cells, on the other hand, are cells that only have 1 of each type of chromosome. They'll be denoted with just the letter N. Haploid chromosomes are going to be things like our sex cells or our gametes. Take a moment here. Why do our sex cells or gamete cells only have 1 set of chromosomes? Our diploid cells are going to have 46 chromosomes each, but why do our gametes only have 23? So not only do the chromosomes occur in pairs, but the genes occur in pairs as well. Genes are going to be a specific segment of the chromosome. If the human genome is a textbook with different chromosomes as the chapters, then the genes are the individual sections of each chapter. Each gene encodes for information on how to make that necessary proteins that allow us to function in the way that we do. And this is how you are able to contain information from both parent. One term that we're going to use a lot is the allele. Allele is the term that we use to describe for alternate forms of the same gene. So both chromosomes may code, or the same gene on both sides may code for color of eyes. Whether they code for brown eyes or blue eyes, those are different alleles. A single gene may encode for the color of the eye, but the color will vary depending on if you have an allele for blue eyes or an allele for brown eyes. Alleles for a gene will always be located on the same location of homologous chromosomes. So this right here could be the gene for eye color. The gene over here could be for finger length. The gene right here may be for how hairy your toes are. So all of that is going to be encoded for in the same locations on our genes. All right, and that's it for the form of chromosomes.